Hi there, how are you? Welcome to another biology class. Today we will continue what we have started last time, part 3 of the discussion on chloroplast. Before we start, make sure you have subscribed to this channel and please be kind to like and share this video. Alright then, let us begin. Green algal-derived chloroplasts. Green algae have been taken up by the euglenids, chlorarachniophytes, a lineage of dinoflagellates, and possibly the ancestor of the cash lineage, cryptomonids, alveolates, stramenopiles and haptophytes, in 3 or 4 separate engulfments. Many green algal-derived chloroplasts contain paranoids, but unlike chloroplasts in their green algal ancestors, storage product collects in granules outside the chloroplast. Euglena, a euglenophyte, contains secondary chloroplasts from green algae. Euglenophytes. Euglenophytes are a group of common flagellated protists that contain chloroplasts derived from a green alga. Euglenophyte chloroplasts have 3 membranes. It is thought that the membrane of the primary endosymbiont was lost, leaving the cyanobacterial membranes, and the secondary host's phagosomal membrane. Euglenophyte chloroplasts have a paranoid and thylakoid stacked in groups of 3. Photosynthetic product is stored in the form of perimylon, which is contained in membrane-bound granules in the cytoplasm of the euglenophyte.
Chlorarachniophytes. Chlorarachniophytes are a rare group of organisms that also contain chloroplasts derived from green algae, though their story is more complicated than that of the euglenophytes. The ancestor of chlorarachniophytes is thought to have been a eukaryote with a red algal-derived chloroplast. It is then thought to have lost its first red algal chloroplast, and later engulfs a green alga, giving it its second, green algal-derived chloroplast. Chlorarachniophyte chloroplasts are bounded by 4 membranes, except near the cell membrane, where the chloroplast membranes fuse into a double membrane. Their thylakoids are arranged in loose stacks of 3. Chlorarachniophytes have a form of polysaccharide called chrysolaminerin, which they store in the cytoplasm, often collected around the chloroplast paranoid, which bulges into the cytoplasm. Chlorarachniophyte chloroplasts are notable because the green alga they are derived from has not been completely broken down. Its nucleus still persists as a nucleomorph found between the second and third chloroplast membranes, the periplastid space, which corresponds to the green alga's cytoplasm.
Prasinophyte-derived dinophyte chloroplast Lepidogenium virid and its close relatives are dinophytes, see below, that lost their original peritinin chloroplast and replaced it with a green algal-derived chloroplast, more specifically, a prasinophyte. Lepidogenium is the only dinophyte that has a chloroplast that's not from the rhodoplast lineage. The chloroplast is surrounded by 2 membranes and has no nucleomorph. All the nucleomorph genes have been transferred to the dinophyte nucleus. The endosymbiotic event that led to this chloroplast was serial secondary endosymbiosis rather than tertiary endosymbiosis. The endosymbiont was a green alga containing a primary chloroplast, making a secondary chloroplast. Red algal-derived chloroplasts. Cryptophytes. Cryptophytes, or cryptomonids, are a group of algae that contain a red algal-derived chloroplast. Cryptophyte chloroplasts contain a nucleomorph that superficially resembles that of the chlorarachniophytes. Cryptophyte chloroplasts have 4 membranes, the outermost of which is continuous with the rough endoplasmic reticulum. They synthesize ordinary starch, which is stored in granules found in the periplastid space, outside the original double membrane, in the place that corresponds to the red alga cytoplasm. Inside cryptophyte chloroplasts is a paranoid and thylakoids in stacks of 2. Their chloroplasts do not have phycobilisms, but they do have phycobilin pigments which they keep in their thylakoid space, rather than anchored on the outside of their thylakoid membranes. Cryptophytes may have played a key role in the spreading of red algal-based chloroplasts.
Haptophyte shaptophytes are similar and closely related to cryptophytes or heteroconophytes. Their chloroplasts lack a nucleomorph, their thylakoids are in stacks of 3, and they synthesize chrysolaminerin sugar, which they store completely outside of the chloroplast, in the cytoplasm of the haptophyte. Heteroconophytes, stramenopiles, the photosynthetic pigments present in their chloroplasts give diatoms a greenish-brown color. The heteroconophytes, also known as the stramenopiles, are a very large and diverse group of eukaryotes. The photoautotrophic lineage, Acrophyta, including the diatoms in the brown algae, golden algae, and yellow-green algae, also contains red algal-derived chloroplasts. Heterocone chloroplasts are very similar to haptified chloroplasts, containing a pyrenoid, triplet thylakoids, and with some exceptions, having four-layer plastinic envelope, the outermost epiplastid membrane connected to the endoplasmic reticulum. Like haptophytes, heterocontophytes store sugar and chrysolaminar granules in the cytoplasm. Heterocontophyte chloroplasts contain chlorophyll and, with a few exceptions, chlorophyll C, but also have carotenoids which give them their many colors.
Apicomplexans, chromerides, and dinophytes. The alveolates are a major clade of unicellular eukaryotes of both autotrophic and heterotrophic members. The most notable shared characteristic is the presence of cortical outer region alveoli sacs. These are flattened vesicles, sacs, packed into a continuous layer just under the membrane and supporting it, typically forming a flexible pellicle, thin skin. In dinoflagellates they often form armor plates. Many members contain a red algal-derived plastid. One notable characteristic of this diverse group is the frequent loss of photosynthesis. However, a majority of these heterotrophs continue to process a non-photosynthetic plastid. Apicomplexans. Apicomplexans are a group of alveolates. Like the helicosporidia, they're parasitic, and have a non-photosynthetic chloroplast. They were once thought to be related to the helicosporidia, but it is now known that the helicosporida are green algae rather than part of the cash lineage. The apicomplexans include Plasmodium, the malaria parasite. Many apicomplexans keep a vestigial red algal-derived chloroplast called an apicoplast, which they inherited from their ancestors. Other apicomplexans like Cryptosporidium have lost the chloroplast completely. Apicomplexans store their energy in amylopectin granules that are located in their cytoplasm, even though they are not photosynthetic. Apicoplasts have lost all photosynthetic function, and contain no photosynthetic pigments or true thylakoids. They are bounded by four membranes, but the membranes are not connected to the endoplasmic reticulum. The fact that apicomplexans still keep their non-photosynthetic chloroplast around demonstrates how the chloroplast carries out important functions other than photosynthesis. Plant chloroplasts provide plant cells with many important things besides sugar, and apicoplasts are no different. They synthesize fatty acids, isopentenyl pyrophosphate, iron-sulfur clusters, and carry out part of the HEM pathway. This makes the apicoplast an attractive target for drugs to cure apicomplexan-related diseases. The most important apicoplast function is isopentenyl pyrophosphate synthesis. In fact, apicomplexans die when something interferes with this apicoplast function, and when apicomplexans are grown in an isopentenyl pyrophosphate-rich medium, they dump the organelle. Chromerides. The Chromerida is a newly discovered group of algae from Australian corals which comprises some close photosynthetic relatives of the apicomplexans. The first member, Chromerivlia, was discovered and first isolated in 2001. The discovery of Chromerivlia with similar structure to the apicomplexans provides an important link in the evolutionary history of the apicomplexans and dinophytes. Their plastids have four membranes, lack chlorophyll C, and use the type II form of rubisco obtained from a horizontal transfer event. Dinophytes as dinoflagellates are yet another very large and diverse group of protists, around half of which are, at least partially, photosynthetic. Most dinophyte chloroplasts are secondary red algal-derived chloroplasts. Many other dinophytes have lost the chloroplast, becoming the non-photosynthetic kind of dinoflagellate, or replaced it through tertiary endosymbiosis, the engulfment of another eukaryotic algae containing a red algal-derived chloroplast. Others replaced their original chloroplast with a green algal-derived one. Most dinophyte chloroplasts contain form II rubisco, at least the photosynthetic pigments chlorophyll, chlorophyll C2, beta-carotene, and at least one dinophyte unique xanthophil, pyridinion, dinoxanthin, or diadinoxanthin, giving many a golden brown color. 52-49. All dinophytes store starch in their cytoplasm, and most have chloroplasts with thylakoids arranged in stacks of 3. Serratium furca, a pyridinin-containing dinophyte. 56. The most common dinophyte chloroplast is the pyridinin-type chloroplast, characterized by the carotenoid pigment pyridinin in their chloroplasts, along with chlorophyll and chlorophyll C2. Pyridinin is not found in any other group of chloroplasts. The pyridinin chloroplast is bounded by 3 membranes, occasionally 2, having lost the red algal endosymbiont original cell membrane. The outermost membrane is not connected to the endoplasmic reticulum. They contain a pyrenoid, and have triplet stacked thylakoids. Starch is found outside the chloroplast. An important feature of these chloroplasts is that their chloroplast DNA is highly reduced and fragmented into many small circles. Most of the genome has migrated to the nucleus, and only critical photosynthesis-related genes remain in the chloroplast. The pyridine chloroplast is thought to be the dinophyte's original chloroplast, which has been lost, reduced, replaced, or has company in several other dinophyte lineages.
That is all for today, folks. One last thing. Before you go, make sure that you have subscribed to our YouTube channel and be kind to like and share this video. This video is edited by Brooke Solomon, MSc in Genetics. I will see you in the next video. Goodbye.